In the scene Mr. DNA, our group encountered many mistakes within the movie. First of all, it would be extremely difficult to keep intact a DNA strand for over a million of years. Furthermore, scientists could have possibly mixed up the dino DNA with the mosquito DNA in the amber. The dinosaur's DNA extracted from the amber would have only contained a few cells. Not only would it be lacking cells, but how would scientists know that the DNA in the amber was really the dinosaur's DNA? In the movie, when the dinosaur DNA was revealed, there were many missing sections in the DNA. The scientists then used frog DNA to replace the missing sections. First of all, in order to perfectly clone the DNA in the amber must be perfectly preserved. But in the movie, it was not. Second, frog DNA is not the same as dinosaur DNA. Bird DNA is more similar to dinosaur DNA than frog DNA. If even 1% of or one base of the DNA is missing, the scientists would be unable to produce a dinosaur. In order to perfectly clone a dinosaur, you need to close the gaps in the DNA strand. In addition, how would scientists even figure out the amount of missing DNA? The gap can vary from 3 bases to 500 bases of DNA. Plus, how would the scientists figure out how many chromosomes are in a dinosaur? As you can see in the movie, there were many mistakes within the scientific studies. Hello, doctor. Doctor.
Who are you? So what do you want?
Oh, I'm just here to ask you, how did you make that dinosaur?
Come over, come here. Come. You see, I use my supercomputer here, and then we use the DNA and we copy stuff, okay?
Yeah.
The basic method was to identify individual dino genes by placing the dino DNA fragments alongside similar sequences within known bird genomes. Step 1. Pick out one fragment and write down a sequence of 15 bases. Have the computer look through all the other fragments to see if you can find the same sequence. The 2 identical fragments would be almost certain to come from the same place on the genome. On the computer screen, you can place those 2 fragments side by side. You can use this to match the ends of new piece of some other fragment. Step 2. You can then sort through the rest of the fragments for more repetitions. In order to have a chance of putting the whole genome together, you need 3 or more original copies chopped up in different ways. Then your supercomputers will search through the some 100 million scraps of DNA to see which fragments match up. Step 3. You would need to have both sets of chromosomes of the parents with intricate detail. Tell the computer to look for links of repetitive sequences that could possibly be telomeres. You must get the repetitive sequence right, or else your reconstructed chromosome genetic appearance could look right, but the physical arrangement would be all wrong. Step 4. You would patch together all the fragments of dinosaur DNA you've managed to collect to find missing fragments. The computer may provide you with several possible ways of patching some fragments, but there is no indication which way is right. You would then go to the birds and pick a variety of birds, including primitive ones. Also include a variety of other birds with varying degrees of closeness to the rarities for comparison. Sequence completely the genomes of several birds of as many species as possible. You then compare the dinosaur DNA fragments with the bird sequences. Look for similar or identical sequences. If you can find a close cousin and it's sufficiently long to be essentially unique, you could use that bird genome as both a guide to where the corresponding bit of dinosaur gene might get along with the dino genome and as a model for filling in missing pieces in dino gene.
Hey, doctor. Hey, doctor.
Doctor.